So I think this is, I love the title of this session, so thank you, Rob, for inviting me. I'm honored to get to moderate this session entitled Microbes and Cancer. And we have this distinguished panel of folks who will be talking. So I'll introduce our first speaker, Rick Peake, if you don't know him. It's fantastic, and I just have to say he was my first mentor as an independent faculty member at Vanderbilt 10 years ago, and you couldn't ask for a better mentor, colleague, and now friend. So I know I am one of many who can say that about Rick Peake. He is a professor of medicine and chief of gastroenterology at Vanderbilt, and he is really a world leader in H. pylori-induced gastric pathogenesis. So without further ado, Rick.
All right, well, Mira, thank you for that very undeserved introduction. But I also wanted to thank Rob and Juha for the incredible invitation. I'm humbled by it. But at the same time, over the last 24 hours, I've been inspired by what I've heard about the passion that the people in this room have for this disease. And so what I wanted to do is discuss a little bit of data from our lab, but also from many other labs, including my co-speakers in this session, that have really tried to look at how H. pylori infection can drive and initiate the progression to gastric cancer, and what I consider to be very disruptive in enabling relationships with its host. So essentially, all persons that are colonized with H. pylori develop coexisting gastric inflammation, and this, over a period of decades, really incurs the strongest known risk for gastric cancer. And in fact, the attributable risk for distal gastric cancer by H. pylori is about 90%. The other problem is that about 4.4 billion people worldwide harbor this infection. Adenocarcinoma of the stomach, as we know, is a huge problem. It's the third leading cause of cancer-related death worldwide, and approximately 800,000 people will succumb to this disease this year. We know a lot more about the molecular features of gastric cancer thanks to great work by the Cancer Genome Atlas Network, in which they used a multitude of molecular techniques to classify gastric cancers into 4 predominant subtypes. But in every region of the stomach, chromosomal instability tumors that are tightly linked to intestinal type histology predominate. Dr. Correa has been mentioned many times during this meeting because he identified this very well orchestrated series of histologic steps from normal mucosa to gastric cancer years prior to the discovery of H. pylori. Thanks to his work, we now know that H. pylori is the primary initiator of this cascade. And as we heard yesterday, there are many studies now to show that if you eliminate H. pylori, you can subsequently reduce the risk of developing gastric cancer. So why don't we indiscriminately test and treat people universally? Well, one problem is that only a minority of individuals who are colonized will ever develop gastric cancer. And I think this has put an unusual burden on investigators because we want to understand not only the mechanisms through which H. pylori leads to gastric cancer, but we want to look at different categories to see if we can identify biomarkers or predictors that can put patients at high risk. We also have learned over the last 2 decades that you can't conveniently study factors that are in these different domains in isolation. You really have to understand specifically how they interact with each other to understand the full combinatorial effect H. pylori has on this disease. So in our lab, we wanted to approach this and start to build a model of pathogenesis, incorporating elements from these different domains. And we asked a very simple question, and that was whether dietary components can change the balance between H. pylori's behavior as a commensal or as a pathogen, and if this happens by direct modification of microbial virulence. There are different dietary elements that are associated with gastric cancer. We are learning a lot more about the molecular features of things like high salt. Tim Cover at Vanderbilt has shown that high salt can actually increase the virulence capacity of the bacteria. It's not necessarily just incurring damage to the gastric mucosa. But the dietary element that we've chosen to focus on is iron deficiency. So worldwide, iron deficiency is a very significant problem. It affects 30% of the world's population. H. pylori is a strong risk factor for iron deficiency, and concomitantly, iron deficiency is linked with both premalignant and malignant lesions of the stomach. To address this, we had developed a model in our lab of H. pylori-induced gastric cancer using Mongolian gerbils. And through a series of experiments, we were now able to induce both gastric dysplasia and gastric carcinoma in gerbils infected with H. pylori, which occurs in a relatively short time frame, around 8 to 12 weeks. So we did a very straightforward experiment now that we had this animal model up and running. And that was to place animals on an iron normal diet or an iron deficient diet. And then we infected them with H. pylori and looked at the development of gastric dysplasia in yellow and gastric cancer in red. And we saw a very striking finding. We found that if animals were iron deficient when they were infected, their risk of developing dysplasia and cancer was twice as high as if the animals were iron replete. We've now gone on to replicate this in other models of H. pylori-induced injury. We can infect C57 black 6 mice, iron normal and iron deficient, and we see the same phenomenon. More injury if the animals are iron deficient. And we've also done this in a mouse model that has a higher predisposition to gastric cancer. And these are NSCAS mice. So the pattern is the same. So we'd identified a very interesting phenotype. But now we wanted to delve into the mechanisms that underpin this. So one way to do this is to use discovery-based techniques. And so we isolated H. pylori strains from iron normal and iron deficient gerbils, and then we performed whole genome sequencing on these isolates to determine genes that may be mutated under conditions of iron deficiency. And we found that there was a very small workable group of genes that were altered. One of these was in a microbial gene called FIR, which was important because FIR is one of the master regulators of iron-regulated genes in bacteria. But one of the beautiful things that I've been able to do over the years is collaborate with individuals that are much smarter than me and much more driven than me to take our findings in the lab into human populations. So we've had a great opportunity to work with Keith Wilson and Costanza and Blanca Piazuela and Dr. Correa and look to see if any of our findings in the lab can really be replicated in human populations. So in Colombia, almost all the population is infected with H. pylori. But my co-investigators really identified a striking paradox in that on the coast, the risk of gastric cancer is very low compared to levels that you see in the mountains. And in fact, this is a 25-fold difference. And this allows huge opportunities to look at differences in diet, differences in microbial virulence, differences in human genetics. So we simply wanted to determine whether the findings that we identified in our animal model could be replicated in humans. And in fact, we found that this FIR mutation, which is shown in red, was significantly more frequent in individuals that resided in the mountains who had high-risk lesions, atrophic gastritis, intestinal metaplasia, and dysplasia. Well, what other targets could we go after to really understand the pathogenesis at a molecular level? Well, one gene that really caught our eye was a gene called CagY. And one of the most intensively studied virulence factors within H. pylori is a pathogenicity island called the CAG island. And about 60% of H. pylori strains in this country harbor this island. And when the bacteria bind to epithelial cells, this island forms a needle-like structure and it will inject protein called CagA into the host epithelial cell. And once it's inside the cell, CagA can interact and aberrantly activate many pathways, all of which have been shown to be mutated in gastric cancer. And in fact, it can activate beta-catenin. My co-speaker, Manuel Amieva, has also shown that if you place CagA by itself in epithelial cells, this changes the biology of that particular cell. So in this movie, focus in on the green cell. This is a CagA-GFP construct that's been placed in one cell. The cells around it have been pseudocolored to allow for an easier visualization of their movement. And if you follow the behavior of the cell over time, you can see that it moves much farther and much faster in this monolayer than the other cells. And this is a phenotypic characteristic of a cancer cell. So this is a schematic of the CAG island that translocates CAG-A into host cells. CAG-Y is a structural component of this island. So we want to determine whether this mutation that we had found really had any bearing on assembly of this virulence factor. And so you can take H. pylori strains from iron normal gerbils, you can add them to gastric epithelial cells, and they will form the secretion needle. And the secretion apparatus is injecting CAG-A from the bacteria into the host cell. But look what happens when you look at iron deficient strains. Many more of these secretion pili form. All of them are functional, all injecting CAG-A. Well, again, we wanted to move and validate our findings in human populations. So we had access to human samples from the high and low risk region, and we were able to show that indeed in individuals that had the most severe lesions in the stomach, these individuals had the lowest concentrations of iron. But we also had access to the H. pylori strains that infected these individuals, and we were able to show that strains in yellow from people with low iron concentrations actually were more pathogenic. They induced more cytokine production when we looked at their co-culture with gastric epithelial cells. So one of the people that I had the great fortune to train with, Martin Blazer, is a microbial infectious disease expert. And he always said, with a chronic pathogen, no matter what you find in the lab, you've got to think about what advantage it provides to the bacteria, not what element of damage it's really inducing in the host. So why would a chronic pathogen want to inject an oncoprotein into host cells to potentially eliminate the host? Well, in one of our great collaborations we've had with Manuel, he had developed a transwell system in which he could place human epithelial cells in a transwell, and then he could add H. pylori to the top of these cells. And when he added a wild-type strain of H. pylori, the bacteria in green grew well, they replicated, they formed microcolonies very easily. But if you mutated the CAG-A gene in this strain, all of a sudden it lost the ability to colonize epithelial cells. But he could completely restore this phenotype by adding iron back to the mutant. So this raised the hypothesis that one function of CAG-A from the bacterial perspective is to acquire iron from its host. So in a series of very dedicated and long experiments, Manuel's group used biotinylation assays, and they were able to show that indeed, when H. pylori injects CAG-A into an epithelial cells, one of the downstream consequences is that an iron source, transferrin, will move through the cell from the basolateral surface to the apical surface of the cell where the bacteria reside. And the bacteria can then use this as an iron source. And if you place the bacteria under iron deficient conditions, its response is to inject more CAG-A into the cell to get more of this iron source to allow it to survive. So are there other elements that we need to add into this model, into this equation about how H. pylori may increase cancer within an iron deficient environment? Well, yes. And one of the things that we looked at really stemmed from a clinical paper. So we heard yesterday mention of a trial called the Shandong trial in China, in which investigators treated individuals with antibiotics, and then they followed them out to 15 years. And then they looked to see if those individuals had a reduced risk for cancer. And indeed, the individuals that were treated for H. pylori had a significant reduction in the subsequent risk of developing gastric cancer. But in this article, they also looked at the percentage of individuals that were eradicated for H. pylori at the 15-year time point. And among individuals who had received antibiotics, less than half were successfully cleared. So this raised a hypothesis that antibiotics may be impacting other microbial communities besides just H. pylori in the stomach. So specifically, we wanted to know what are the roles of non-H. pylori constituents of the gastric mucosa in disease that may be enhanced by iron deficiency. So if you look at data that's been accumulated on the effect of the gastric microbiome in gastric cancer, it's less robust than what's been done in the intestinal microbiome. In humans, in general, in individuals who are infected with H. pylori, their microbial diversity in the stomach is less compared to uninfected individuals. In mice, you can manipulate the gastric microbiota simply by infecting with H. pylori. And when you do that, you see a contraction of the Bacteroides, you see an expansion of the firmicutes. Does this have anything to do with disease? Well, in mice it does. This is a study that was published by Jim Fox and Tim Wong, in which they used germ-free mice that were then monocolonized with H. pylori. And this is shown in green. And when they monocolonized these germ-free animals with H. pylori, 80% of those animals developed no evidence of neoplasia in the stomach. And the ones that did had low-grade lesions. In contrast, mice that had a conventional microflora that were then infected with H. pylori, 80% of those animals developed high-grade neoplasia, suggesting that the gastric microbiota may collaborate with H. pylori in accelerating the progression of disease. There's also data from computational biology studies that have supported this hypothesis. So this is an article that was published in which the investigators developed a technique where they could look at all published cancer genomes and they could identify bacterial DNA fragments. And when they did that, they found that the cancer that had the second highest amount of bacterial DNA was gastric adenocarcinoma. But when they looked at the actual sequences, it wasn't H. pylori DNA. It was DNA from other bacteria. And the highest reads were from Pseudomonas. So I think this has forced us to alter the model in which we think about how H. pylori may cause gastric cancer, in which early on there are very intense interactions between H. pylori and its host. Robust inflammation develops. Over a series of years, atrophy then develops, and you're allowing other bacteria to colonize. So maybe understanding what these bacterial components are will provide more insight into gastric cancer. So how do you do that? Well, human populations really are the route to pursue. So with all the collaborators that I mentioned earlier, and Jim Fox and Sebastian Serabom, we were able to catalog species in the stomachs of individuals in the high-risk regions of Colombia and the low-risk regions that were only present in those two groups. And you can see there's a very small workable group of bacteria that are either present only in the low-risk or only in the high-risk individuals. What about other locations? Well, this study was published in Gut, and it was a study from 2 sites in China in which they looked at the microbial composition in the stomach in individuals that either had normal gastritis or gastritis alone, atrophy, intestinal metaplasia, and gastric cancer. And then when they looked at species richness, it decreased as you progressed to gastric cancer. They also did a very interesting technique in which they measured the strengths of the co-occurring or co-excluding relationships among bacteria in the stomachs of these individuals. And you can see there's some mildly strong relationships among bacteria in the stomach of individuals with superficial gastritis. But look what you see when you look at these relationships in gastric cancer, reinforcing the hypothesis that these bacterial species may really collaborate with each other to promote disease. And these were some of the species that were present more commonly in individuals with gastric cancer. Can these results be extrapolated to other populations? In fact, yes. A study from Portugal showed very similar findings in that there was a reduction in gastric microbial diversity in the stomach in individuals who had gastric cancer, and microbial dysbiosis scores were increased in those individuals as well. So how do we bring this back to our model that incorporates iron deficiency? Well, we wanted to look at the microbial communities in our infected gerbils. And one question that we wanted to initially answer was, well, do different iron levels affect the bacterial populations? And in fact, they don't. If you look at the principal component analysis, which reflects community structure, you can see that iron deficient and iron normal bacterial populations almost completely overlap. There are no differences. But if you start to look at the microbial factors, you can see that in gerbils that were infected with a wild-type strain in red, their population structure is very different than either uninfected gerbils in white or gerbils infected with a CagA isogenic mutant. And we've also been able to look at this at a phyla level, showing these differences, and we're now investigating this in more depth. So the final piece in this model is the question of whether location matters in the stomach. Does it matter if H. pylori colonizes the superficial region of gastric glands or other regions that may be more susceptible? And again, with Manuel's help, he had developed beautiful imaging techniques. This is 3D in vivo confocal microscopy. And this is a gerbil stomach that was been infected with H. pylori. And we started to see bacteria that were not only on the surface, but also there were pockets of replicating bacteria that were deep down in the gastric glands. And when we looked at the difference in our iron-depleted and iron-normal gerbils, there were more bacteria that were present in the iron-deficient animals. And these were adjacent to progenitor and stem cell zones. So how do pathogenic H. pylori interact with these susceptible cell populations? Well, again, Manuel's group really led the way. They looked in mice, and they were able to show not only could they identify pockets of replicating bacteria deep down within the gastric glands, but many of the cells that were adjacent to these pockets of bacteria were undergoing proliferation, suggesting that H. pylori may be able to activate proliferation of a stem cell population. So to really pursue this in more depth, we wanted to focus on a specific stem cell population. So we focused on cells that are called LRIG1 positive cells. LRIG1 marks quiescent stem cells in the stomach. It's an inhibitor of EGFR signaling. And the reason that we focused on this stem cell population was we had shown previously that if you infect mice with H. pylori, the number of these stem cells increases. If in an animal model you mutate one copy of APC only in LRIG1 positive cells, all these mice develop gastric premalignant lesions. And finally, moving into humans, we were able to show that indeed, if you look at people that have premalignant lesions, atrophic gastritis, there are many more LRIG1 positive cells shown in brown than what you see in superficial gastritis. So we did a study that we wanted to really determine whether H. pylori could functionally activate the stem cell population. And one way you can do this is by lineage tracing. So Jim Goldenring and Bob Coffey had a mouse model in which you can treat this genetic model with tamoxifen, and it will mark all the cells in the stomach that originate from an LRIG1 positive cell. We then infected those mice with H. pylori. And if you look in the left panel, these are uninfected mice. You see one gland that's populated with these green cells. That means an LRIG1 positive cell gave rise to these cells in this single gland. Look at the level of functional activation we see when we infect with a CAG positive strain of H. pylori. When we knock out CagA and infect mice with this mutant, it really resembles uninfected mice. We were also interested to determine whether H. pylori could activate LRIG1 positive cells to lead to premalignant lesions. And so Jim Goldenring had identified a form of metaplasia called SPEM, which many people, at least in mouse models, feel is in the progression to gastric cancer. And the hallmark of SPEM is to have these pink cells at the base of the gastric glands. So if you look at uninfected mice, we're staining for the SPEM marker in pink. The pink cells are really in the middle of the gastric glands. However, when we infect with our CAG-positive strain of H. pylori, now we see pink cells at the base, and they're co-localizing with green cells, which are marking L-RIG-1 positive cells, indicating that infection with a CAG-bearing strain of H. pylori can induce potentially premalignant lesion originating from L-RIG-1 positive cells. So how do we build this into a model? Well, I think H. pylori is able to colonize microniches within the stomach. And if you find the bacteria in progenitor or stem cell zones, and if it's a pathogenic strain that has a functional CAG island, it can inject CAG-A into these cells. It can activate beta-catenin, it can induce DNA damage, but remember, all this is occurring within a very inflamed microenvironment, full of reactive oxygen and reactive nitrogen species. Over years, atrophy develops. This inhibits acid production. You need acid to absorb iron, and now you've selected or put the bacteria under a stress of iron deficiency, and its response is to inject more of this oncoprotein into the host cell. And we would speculate that more CAG-A in the cells actually can then lead to a modification of the gastric microbiota. And this really is in a feed-forward detrimental loop. So the last thing I'll just mention, are there other technologies that we can use to really delve into molecular mediators of iron deficiency in this model? Yes, so we've used other discovery-based pathways. We've done metabolomics on H. pylori strains from iron-deficient gerbils. We found many different targets that are aberrantly activated. We've done proteomics on the same strains. Many of these pathways are common. So these are the targets that we're now going after. The other thing that I wanted to show you is, working with animals and things like that. Please play. Okay. It would be great if we could follow these animals over a time course, because right now we're really relegated to, doing sacrifices and only looking at one particular time point. So we wanted to develop techniques in which we could use our animal models and follow animals longitudinally. And so we developed a technique using a flexible ureteroscope to do upper endoscopy in gerbils. So this is the first endoscopy that we ever did in a gerbil. We're down in the esophagus. We're about to pop through the lower esophageal sphincter. And when we did this, we hit this massive load of food. And so we didn't really anticipate this. So I had to sort of, go back in time and relearn what I'd forgotten in fellowship. So we fasted the gerbils. We gave them a liquid diet for 2 days. We put them on wire bottom cages so they wouldn't eat their feces. And when we did that, the next time we were able to get a much clearer view of the mucosa and were able actually to take biopsies. So I wanted to go ahead and end. What we've tried to do is incorporate elements of these multiple different domains and put specific titles or names on how these different factors may interact specifically with each other and really impact the development of gastric cancer. So I wanted to thank many people in our lab, but also the wonderful collaborators that I have the great opportunity to work with over the years. And thank you for your attention.